each other, and then the pattern is determined by this pattern of hydrogen bond donors and acceptors. And in the case of xanthine, the pattern of donors and acceptors matches better with cytosine than with thymine, even after that chemical change. So there are chemical changes that can happen to bases that don't directly affect their base pairing. So there's one thing we know is that uracil and thymine have actually the same base pairing preferences. And actually, I'm just going to flip back here to point out that the methyl on thymine is totally separate from the base pairing interface here, and so it does not matter to the base pairing preferences of uracil versus thymine. And actually, in a similar way, you can have methylation of the 5 position on cytosine. So thymine is 5-methyl-uracil, 5-methyl-cytosine here is an analogous base. And so there's no change to the pattern of hydrogen bond donors and acceptors when you put this methyl here. No change to the base pairing interface. And so cytosine methylation happens naturally in many different cells. When it happens, acetylmethionine is the methyl donor that puts the methyl group onto that position. And so in bacteria, the methylation of cytosine, as well as certain positions of adenine, is used to mark self-DNA in the bacteria, as well as in certain cases in DNA repair that we'll discuss later. But it gives essentially it gives the bacterium a chance to mark its own DNA with a mark that's not present on DNA from an invading virus, and that's useful to the bacteria's ability to get rid of viral DNA. In eukaryotes, it does something different. In humans, cytosine methylation occurs mostly at the CpG sites, so it's just a C immediately next to a G. If you think about it, it means on the complementary strand there will be a C and a G as well, and those cytosines tend to be methylated, and their methylation is very important in controlling gene expression. And I think one interesting point about this is the presence of that methyl on a cytosine, so the cytosine 5-methylcytosine, is actually, it is non-base pairing information that's contained in the physical structure of DNA itself that both bacteria and eukaryotes use. So it's something that actually can't be read out by base pairing. Both of these will base pair best with G, but there are actually specialized proteins that can distinguish between these two. So cytosine methylation of this 5-methylcytosine does cause a problem. So I was telling you earlier that cytosine deamination to uracil is a signal that there is a mutation that needs to be corrected because uracil should never be in DNA. Of course, 5-methylcytosine deamination, which happens essentially as readily as a deamination of ordinary cytosine, creates thymine. And there's no way for the cell to know. Simply the presence of thymine in DNA is a problem. So there are still ways that the cell can simply detect a mismatch. So of course, if you have a 5-methylcytosine paired with a guanine, and that 5-methylcytosine becomes a thymine, you now have a thymine-guanine base pair, which is not as favored as the cytosine-guanine base pair, and the cell has a more general mechanism to detect mismatches like this and others. But this very specialized and very efficient repair of uracil in DNA can't actually act in this case. So I mentioned that the cell does have a way to detect cases, sort of any sort of mismatch or problem in the base pairing in the DNA double helix. And one of the places where that's the most important is sort of larger chemical changes to the bases. So we've been talking first about cases where essentially you're changing one very reasonable base into another, but sometimes more dramatic things can happen. One of the most important is a photocyclization reaction that can happen. So this these chemical reactions are the reason that UV light is mutagenic. What happens is that when you have two adjacent thymines are the most favorable for this, but really two adjacent pyrimidines. So these bases will be stacked like this in the double helix, and the two double bonds here, catalyzed by UV light, can undergo two kinds of rearrangements. One is the formation of this cyclobutane thymine dimer, and the other is this photoproduct here. In both cases, the pi electrons from the two adjacent pyrimidines are reacting in order to form a covalent bond between adjacent bases. So this is not something that ever happens in normal DNA as a cell synthesizes it, and in fact, the bonds between these two pyrimidines distort the backbone, they distort the base parent interface, and actually this serves as a complete block to a lot of the enzymes that work on DNA in cells. So this is one sort of example of a sort of a larger kind of damage to the cell, and what the cell essentially has to do to get rid of this is cut this region out of the DNA and repair it. And in fact, that's the cell's approach to many problems in nucleic acids is they see a problem, they try to cut out the damaged strand and repair it. And so that sort of leads into the general question about nucleic acid backbone chemistry. So there are a lot of related chemical reactions that the cell uses to cut the backbone of DNA or RNA, to extend it, or to join two strands together. And essentially all of these things have a single basic chemical reaction that's going on, this transesterification reaction. And so it's definitely worth looking at and learning in detail, because this same basic pattern comes up again and again in molecular biology of DNA and RNA. So in order to cut the backbone here, for instance, an enzyme will position and activate a nucleophile to attack the phosphate and create an intermediate where this phosphate is pentavalent, looks like this. The attacking nucleophile is here, and the electrons are displaced on to make this. And that's resolved when the phosphate kicks off one of these other two substrates. So here, the phosphate kicks off this upper deoxyribose, releasing this and leaving the phosphate group on the 5' end of the cut DNA. And actually, it could just as easily kick off the other side and leave the phosphate with this deoxyribose. But essentially, all of these reactions proceed by the attack of a nucleophile, creating this intermediate, and then kicking off a different substrate. And someone asked earlier about the stability of RNA, and the sort of confusing point that the RNA duplex is more stable, but actually the RNA backbone itself is chemically substantially more fragile. And the reason for this is shown here. So before I showed water sort of positioned by an enzyme acting as a nucleophile to cleave DNA. RNA has a problem that the 2' OH on RNA can be activated itself as a nucleophile. So the RNA actually contains the nucleophile that's going to attack the backbone. This is catalyzed by a base which can deprotonate this oxygen and make it into a good nucleophile. And when that happens, the 2' O attacks, and eventually this 5' end is kicked off. So I mentioned that this could be resolved in either way. And here's a case where the phosphate actually stays on the 3' end and forms this funny structure. It's a 2', 3' cyclic diphosphate on this RNA. And here, there's just a free 5' OH. So this actually is not such a fast reaction at neutral pH and at room temperature. Each bond in each phosphodiester in an RNA backbone has about a 4-year half-life. But this can be really dramatically accelerated, of course, by heat, which accelerates many chemical reactions, by bases, which can activate this 2' OH to serve as a good nucleophile, and also by cations that can stabilize some of the intermediates involved in this process. So RNA in pure distilled water is not that fragile at room temperature, but there is, in the context of living cells, lots of cations around at higher temperatures, or definitely in the presence of base, it's much more fragile. So the same sort of chemistry also underlies adding a nucleotide to the end of a DNA or an RNA. So here, the 3' OH of at the 3' end of a DNA or an RNA strand serves as a nucleophile. So all of the NTPs and DNTPs in the cell are 5' triphosphates like this with 3' OHs. And so what happens is the 3' OH of a DNA or an RNA strand is activated to serve as a nucleophile to attack the alpha phosphate of this DNTP, which then kicks off this inorganic pyrophosphate and forms this bond. So in looking at this, of course, the phosphate here in the backbone came from the alpha, the first phosphate on the DNTP here. And after this reaction, there's a new free 3' OH that is again available to be activated and serve as a nucleophile to attack another DNTP and so forth. And so that's the way you can actually synthesize a long nucleic acid chain. This is the basic mechanism of DNA and RNA polymerase. It's shown in a bit more of a schematic form here with the primer strand, and on that primer strand, the strand that's growing, there's a free 3' OH that attacks a new DNTP, kicks off a pyrophosphate, and has a new 3' OH that can grow again with attacking the first phosphate on this DATP. And of course, when DNA and RNA polymerases are synthesizing this, they do essentially the same chemistry regardless of what the nucleotide is, and the specificity, the way they ensure that they're making a copy of themselves is by monitoring the base pairing of the DNTP with the template strand before they commit to carrying out the chemistry. And of course, the strands of, in a double helix of DNA or RNA are anti-parallel. What that means is that this is the 3' end of the strand that's growing, but here's the 5' end of the template strand, which runs from right to left as it's drawn here. And that's, I think, very important to always keep in mind. That's the way you copy it this way from this strand. And once you say copy this strand, you could then turn around and copy it in this direction on the other strand. And that's really the heart of this idea of copying information in DNA. So DNA is, in some sense, all chemically very similar, which causes some problems when you want to actually determine the sequence of the DNA. There are some minor chemical differences between the bases, and that actually is something that can be exploited in an older technique called Maxim-Gilbert sequencing. Just use some chemical tricks to, say, cleave DNA only at the C's, and then you can tell the positions where it got cleaved, those are C's, and so forth, and it's sort of some complicated and somewhat finicky chemistry. Essentially, all of the modern sequencing technologies instead use base pairing to really read the information of the DNA directly. And so what that requires is some way to monitor which base can and cannot pair with a given point in the DNA, with a given position in the DNA. And using actually a DNA polymerase, the enzyme that the cell uses to synthesize DNA, in order to read out this information. And the Sanger method of sequencing relies on exactly this sort of trick, and the way that they're able to do that is to use a dideoxy nucleotide. And so the idea here is that the 3' OH on the deoxyribose is changed to an H. So you remember that in DNA, when we changed the 2' OH in RNA to a 2' H on DNA, this was no longer able to serve as a nucleophile in attacking the phosphodiester backbone. So likewise, when you change the 3' OH on DNA to a 3' H on a dideoxynucleotide, it's no longer able to act as a nucleophile, and it can't extend, and so it can't participate in this reaction here. So once you've put the dideoxynucleotide into the DNA, it has a 3' H, and that 3' H can't be extended anymore, and the DNA chain has been terminated. And the trick to doing sequencing with dideoxynucleotides is to add just a single one of the dideoxynucleotides, and that way the extension of the chain stops, depending, for instance, if I have only dideoxy GTP in the reaction, then the DNA will stop when it's extending a G, but not at A's, C's, or T's. So that is actually the sort of starting point for this next iClicker question here, which is that if you have this sort of sequence of DNA and you're going to extend this top primer strand in the presence of ATP, CTP, and TTP, so normal deoxynucleotides there, and then only dideoxy-GTP, the question is, which of these products do you turn this template strand into here? And I've colored in orange the nucleotides that are being added. And so I'm going to go ahead and start this and have about a minute and a half to talk amongst yourself about which of these products are going to be formed.
Oh.
It's definitely not A, guys. Don't forget.
Freaking.
I could. We just don't get.
Why do you have 3 What do you have, 30?
That's the one time you say it right.
It looks like it's 6 times.
Is that a thing?
That's why you just take it.
Oh, I do have 3. Where's this coming from? Help me here.
I know a lot of people do that.
Okay, so the next 10 or 15 seconds, if you could wrap up your answers. Okay. So product D is the product that will actually be formed. So the things to keep in mind here, first of all, that the strand that's being synthesized goes from 5' to 3', so left to right on the strand that's being synthesized. So we know that D and E aren't that. The polymerases never add to the 5' end of a growing strand. We're looking at where this reaction stops in the presence of DDGTP. First of all, that means that so the guanine is the nucleotide that's being added, and so the reaction is going to stop when it adds a guanine nucleotide. And so, and secondly, remember that the dideoxynucleotide terminates the chain after it's added. So DDGTP can be attacked just fine. The 5' triphosphate will do that chemistry just fine. But once the G has been added in product D, the reaction stops. So in this reaction, all of the chains would go adding these 5 bases, add the poison guanine nucleotide, and then stop. But if you actually want to do sequencing, that's not exactly what you do. You actually want to add in only trace amounts of the DDNTP because you want to detect not just the first G in your reaction, you want to detect all the Gs. And by poisoning the synthesis with a relatively small amount. If you have 1% DDGTP in the reaction, then you have about a 1% chance of stopping at G's, and you'll actually get some extension products that span a range of different lengths, each stopping at one of these G's. And here's a schematic sort of example that's happening with DDATP, and you can see that when you have the DATP in the reaction as well as the dideoxy analog, you can get both of these products as well as some longer things as well. The example from Leninger, which you can actually do, of course, is set up 4 different reactions. And here, each of these bands represents a single product where chain extension stops. For instance, here in the presence of DDATP, chain extension stops after adding an A to the 3' end of the growing strand here, here, and here. And likewise for where C's, G's, and T's stop the extension of the growing strand. This is what the data would look like from sort of classic Sanger sequencing. We look at this, first of all, by radio labeling your primer, by performing 4 of these reactions, one poison with each of the DNTPs, and seeing at what position each of those poisoned DNTPs stops the reaction. When you actually do DNA sequencing nowadays, first of all, it's typically done as a sort of a commercial service, and they have a trick, essentially, that they use to do this sequencing. And the idea is that instead of doing 4 different reactions, they do a single reaction where they put trace amounts of all 4 DNTPs. And each of those DNTPs is labeled with a different color of fluorescent dye. And so what that means is that for instance, the green DDATP does 2 things. It stops the synthesis after adding an A into the chain, and it dyes the product chain green. And likewise, the red DDTTP stops the reaction after adding a T and dyes those chains red. So in a single reaction, you have 2 things. You have chains terminated at different positions based on what nucleotide they contain at that position, and colored different colors based on the identity of that nucleotide. And that you can then separate out on an instrument. So this does sort of the same thing as gel electrophoresis to separate DNA based on size, but it does it in this capillary and then reads out the colors of those different fluorescent dyes based on their, as they migrate past the detector. And you get this sort of trace, and you can essentially just look on this trace and say T, 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 G, A, and so forth. So this sort of method, this Sanger sequencing, is actually the approach that was used, for instance, for the original sequencing of the human genome, as well as many other large projects. But it's sort of fundamentally limited in that sort of at any one time, in any one reaction, you're sequencing just a single DNA molecule. So you need a lot of that DNA molecule to make all of these different extension products. And it turns out that as you resolve these things, you have a sort of a limited range based on, I told you that you need to sort of poison the reaction with a small amount of DDNTP. You need to separate things out by size, and ultimately what that means is you get 400 or 500 bases of sequence from one of these reactions. You get the sequence of a single DNA molecule. More recently, people have developed what's called next generation sequencing, which works using a similar but slightly different principle. And so here, and this is a figure taken from chapter 9 of Leninger, where this is explained, that instead of doing sort of the full extension, what's done is that a single nucleotide is added in the cycle. And that nucleotide is blocked at the 3' end, sort of like a DDNTP, except instead of just putting an H there, they put a chemical group on there that blocks extension, but so you can come in and cleave off later. And the other thing that they do is the dyes that are attached to these blocked nucleotides can also be cleaved off. So you can do a single round of addition here, where on this sort of example, you've added in a blocked TTP. That blocked TTP is yellow, and you can only add in one nucleotide, so you can't add in the next A because this T is blocked. So you do this, you look at the color of your DNA, and then you do a cycle of cleaving off the dye and the blocking group and do another round of addition. Here, you add in a single A. That A, again, is blocked in the 3' end, so it can't be extended further. In this case, it's dyed blue. You can look at that and repeat, where a series of these colors are going to read out each of the nucleotides as they're sequentially added and then deprotected and allowed to undergo one more cycle of sequencing. And ultimately what that means is you have these DNA molecules arrayed on this little slide here. And after each cycle, each of these DNA molecules, one spot, shows up as a different color. And of course the computer just goes through and looks at the colors of all these spots over all these cycles and reads out the sequences. And when you do this, you can get the sequences of hundreds of millions of DNA molecules at once. And this is the way that people were able to take sequencing the human genome from an immense undertaking to something that people can do, if not routinely, at least commonly, and for many thousands of genomes. So DNA sequencing is sort of one of these technologies that has sort of really changed the way we do molecular biology. And I think the other sort of complementary technology is the chemical synthesis of DNA. And just want to mention this briefly and then finish for today. So the chemical synthesis of DNA proceeds by a reaction. And so we don't need to know the details of this. I wanted to point out principally what's possible and what its limits are, but I did think it was worth pointing out that the fundamental chemistry is actually pretty similar, except this that chemical DNA synthesis goes backwards. The 5' OH serves as a nucleophile to attack this chemically modified, actually it's a phosphite group that undergoes this reaction, and then you add one nucleotide and the chain is extended 3' to 5'. But even here, the basic chemistry of this nucleophilic attack is pretty similar. So DNA synthesis chemically is limited by the efficiency of this chemical reaction, which people have worked very hard to optimize, but is still dramatically much less efficient than the reaction actually carried out by polymerases in cells. And so the limitation is about 99